Assalamu alaikum and hello everyone. My name is Muhammad Farhan Tanvir and I'm a student of Bachelor in Pharmaceutical Technology. And today I am excited to tell you about the journey and future of mRNA vaccines. This presentation is for my critical and creative thinking course MPU 3222. So let's begin. Introduction to mRNA vaccines. Let's start by breaking down what mRNA is. Think of mRNA as a messenger. It's a special molecule in our bodies that carries message from our DNA to our cells, telling them to make certain proteins. In vaccines, mRNA gives our cells instructions to make a harmless piece of virus. This prepares our immune system so that if we meet the real virus, our body already knows how to fight it. And the best part, no live virus is involved, making it a safer and quicker way to create vaccines. So let's go to the slide number 3, the timeline of mRNA technology development. Here we will discuss four timelines: the 1960s discovery of mRNA's role in cells, the 1990s when early experiments with mRNA as a potential medical tool was conducted, 2000s is the timeline 3, key breakthroughs by Catalin Carrico and Drew Weissman. And the final, we will discuss about COVID-19 in 2020, rapid development of mRNA vaccines by Pfizer, BioNTech, and Moderna. Okay, let's move to the next slide. The timeline of mRNA technology development, we'll talk about 1960s discovery of mRNA's roles in cells. mRNA discovery 1961. Scientists, including Jacob and Monod, identified mRNA as a genetic messenger, and they found it was a messenger carrying instructions to create proteins. The second point is role of mRNA. Think of mRNA as a blueprint that tells cells what to build. This was a huge discovery for biology. And the third one, filling the gap. This discovery bridged a missing link in genetics and cell biology. And before that, people didn't know why the information was transferred from DNA to cells. And after the discovery of mRNA, they finally understood what's behind the transfer. So let's move to the next slide, which is about 1990s early experiments with mRNA as potential medical tool. Challenges. Early synthetic mRNA was seen as foreign by the body, triggering immune responses. The body saw the lab-made mRNA as foreign, so it triggered an immune response, causing various types of problems in the body. And the second point, the key researchers Catalin Carrico and Drew Weissman saw that mRNA had big potential for vaccines if they just could stop the immune system from attacking it. And they made a breakthrough. They modified mRNA to reduce immune reactions, building a base for mRNA technology. And this breakthrough set the stage for mRNA to be safely used in vaccines. We will discuss that in later. Okay, the next slide. The 2000s. Key breakthroughs by Catalin Carrico and Drew Weissman. The chemical modifications. They found ways to change the mRNA so it could enter cells without causing a reaction from the immune system. And they published research in 2005. In their work, they showed that modified mRNA could safely deliver instructions to cell, which got biotech companies interested. And the final vaccine foundation. This was a turning point. It meant that mRNA could be used to make safe and flexible vaccines. So let's move to the next slide. Development of mRNA vaccines for COVID-19. At first, there was urgency, global pandemic uh, triggered the urgency for mRNA vaccine development and research. And then second, Pfizer and BioNTech and Moderna created vaccines in record time and they make a record rapid development. And they used genetic blueprints, like they used SARS-CoV-2 spike protein sequence for immune response adjustment. And they got the emergency approval, the first mRNA vaccines authorized for public use in 2020. And they finally achieved a milestone and proved that mRNA's potential for rapid, adaptable vaccines development. So now let's discuss how mRNA vaccines work. In this picture, you can see that there is a spike protein. In the first picture, you can see there is a spike protein of a COVID-19 virus. And the second picture, you can see mRNA with instructions for making the spike protein is developed in a lab. The same exact spike protein from COVID-19 virus is developed in the lab, which is not harmful. And they finally make the mRNA vaccines. And after pushing the vaccine in the human body, the mRNA enters the cell and make spike protein like the COVID-19, as you can see in the picture 4. And in the picture 5, you can see spike proteins are recognized by the immune system, which produce specific antibodies against the COVID-19 virus. And the picture 6 is showing if you are infected with the COVID-19 virus, antibodies bind to virus and stop it from replicating. And thus, mRNA vaccine instructs cells to produce a harmless piece of virus protein. This protein triggers an immune response, creating antibodies. Let's move to the next slide. The benefits of mRNA vaccines is quick to develop because they use genetic info. mRNA vaccines are faster to make than traditional vaccines, and it is safer since no live virus is used. There is a no risk of getting infected from the vaccine itself. Point 3, it's easy to adopt. mRNA vaccines can be updated fast to handle new virus strains or other health threats. The point 4 is simple to produce. They are very easier to manufacture, which means they can be produced on a large scale. And the final future potential. mRNA might help treat other diseases like cancer and genetic disorders in the future. So the next slide shows the global impact and success of mRNA vaccines. The success of mRNA vaccines has made a huge difference around the world. The graph shows the reduction in COVID-19 cases and hospitalizations per 100,000 people over time, following the rollout of mRNA vaccines in 2021. The y-axis of the graph represents the number of cases and hospitalizations per 100,000 people, while the x-axis represents months from January to November 2021. And there is two lines displayed, one is yellow and one is orange. The COVID-19 cases is the yellow line with circles, starts at around 100 cases per 100,000 people in January and gradually decreases each month, reaching near zero by November. And COVID-19 hospitalizations is the orange line, begins at approximately 20 hospitalizations per 100,000 people in January and also declines over time, nearly reaching zero by November. The trend indicates a steady decrease in both cases and hospitalizations, with a more significant drop in cases, suggesting that impact of mRNA vaccine in reducing COVID-19 spread and severity. The next slide is showing the future evolution of mRNA technology. Point number 1, expanding to other diseases. The success of mRNA in COVID 19 has led to its exploration for more diseases like flu, HIV, and Zika, showing its adaptability. 2. Cancer treatment. mRNA technology allows for personalized cancer vaccines targeting specific cancer cells and boosting immune responses. 3. Gene therapy potential. mRNA offers a way to treat genetic disorders by providing cells with instructions to correct genetic issues at their source. 4. Rapid response in future crises. The flexibility of mRNA enables quick vaccine development for emerging diseases, aiding in faster pandemic responses. 5. Less expensive and accessible. mRNA vaccines are quicker and cheaper to produce, making them more accessible globally and cost effective. So let's do the next slide. So we reached the conclusion here. The potential for mRNA extends beyond COVID 19 to new treatments and vaccines for other diseases. And mRNA vaccines have transformed the landscape, and their adaptability opens doors for future breakthroughs in healthcare. And to wrap up, we can say mRNA vaccines have completely changed how we make vaccines. They are fast, safe, and flexible, opening up tons of new possibilities in healthcare. And this is just the beginning. We may see various improvements in future. So, thank you all. Thank you so much for your attention. I hope you enjoyed learning about the incredible story and future of mRNA vaccines. Goodbye.